Learning objectives include Western blotting. Western blotting is another serological technique that could be used for microbial identifications. We know that various organisms have common proteins in them, but certain proteins are specific to every single species of an organism or microorganism, and those proteins could be targeted by serological methods. So in this Western blotting, what is done basically is that you take all the antigens, extract the antigens from a single colony of a bacterium, and then you separate those proteins through electrophoresis. And as we know that proteins could be separated based on their molecular weight through the gel. When these proteins get separated, we can transfer these proteins onto another membrane called nitrocellulose membrane by putting high salt and that soaking the gel with the high salt, keeping the filter on the gel, and then that filter is then stacked with lots of paper. And by the wick action of this paper, the high salt is, with the capillary action, is sucked in through the gel. And when high salt moves from the bottom, from the container into this stack of papers, it takes all the proteins from the gel onto the nitrocellulose filter or membrane. And this transfer is exactly the same as we originally separated the proteins. So the proteins remained separated in their individual bands. And then a specific protein that we want to detect could be detected by the use of antibodies, the same method as we used in ELISA. So the antibody has to be labeled with some probe. And then we would allow the filter that has the bands on the bands on the paper, the nitrocellulose filter. We would allow the antigen-antibody interactions to take place, and then we would have a detection system by which we detect the specific band if that antigen is there. For example, after transferring the proteins from the gel onto the filter, we take the filter and then add labeled antibodies into a tube like this. And then we let it roll for 30 minutes, an hour. And then these antibodies are taken away, washed out. Those unbound antibodies are washed out. And then if, let's say, this is a radioisotope, this probe was a radioisotope, we can expose the gel or the filter in this case with X-ray film. And because radioisotopes, they could be detected by the X-ray film, and these images, wherever that protein is there that interacted with our specific antibodies that have a radioisotope probe, it would make a band there. So this is an X-ray film that is basically showing different bands. And that the presence of these bands basically tells us that the protein was there. And the protein, we know it came from a specific microorganism. And that is how we can imply a Western blotting, one of the modified version of a serological technique. In summary, proteins could be separated by electrophoresis, and those proteins could be blotted onto a filter paper. That filter paper could be soaked with specific antibodies, and those bands that interact with those antibodies could be detected.